Okay, let's first of all, using the slides to do a little bit of brief review what we have. Okay, so we talk about the two laws of thermodynamics. The first law is energy not created or destroyed is always transferred. The second law is the entropy going to be increased, which means the disorder will be increased in the system. Then we draw the ATP structure on the blackboard, and we talk about the ATP is the energy currency. And then we started to talk about this. This is a gas equation. You can understand it's a free energy reactions. We have a delta G. We always want a delta G to be negative because the reaction is spontaneous. If it's positive, then it's not spontaneous. So then we explain to you what is delta G, what is delta H, T, and delta S in here. And then we have this equilibrium chemical reaction, which is AB becomes C and D. Then the calculation of the K equilibrium, which is equals C and D divided by A and B. Then we get this standard calculation of the delta G. Now remember, I think I draw on the blackboard on the lab section also. This is minus. So this KEQ here needs to be this one is a positive. If it's this positive, which means delta G is negative. We want it to be negative because this reaction could be processed spontaneously. That's why it came out with KEQ with more than 1, which is delta G is negative. We call it an exogonic reaction and the endogonic reaction, which means KEQ is less than 1.0 and delta G is positive. And then we mentioned about this also using this mechanism to talk about ATP generated during the metabolic process. And we basically talk about here is a substrate level phosphorylation. Now why it is a substrate level phosphorylation will happen? Because ATP itself can hydrolyze and become AMP and ADP. Their phosphate transfer potential is 30-45. And there are some chemicals, typically is from glycolysis, we will mention real quick. They all have a higher phosphate transfer potential than ATP. So that's why they can initiate substrate level phosphorylation. But there are some other chemicals during the glycolysis. It sounds like, when you look at the letter, it sounds like it can do phosphate transfer, but unfortunately, they cannot do because their phosphate transfer power is less than ATP itself. So then we mentioned about another mechanism before we talk about chemosmosis, which is oxidation reduction reaction. Then we're using this slides to talk about what is oxidation, loss of electron, what is reduction, gain electron. Then I say the easiest way to do is you just remember the GRE exams as a student go to graduate school. And then we talk about here, which is oxidized products, reduced products, oxidation reagents, and the reducing reagent. We will mention this again during the fermentation. And then we talk about the electron transport chain. Then I talk about is the order. Why the order is like that? Because we're using a two-half reaction. And the more positive voltage value indicate it's a good electron acceptor, and the more negative indicate is an electron donor. So it is always from a donor to acceptor. So that's why from the top to the bottom, this is basically is chemical compounds arranged in the electron transport chain. So here we also talk about that is more negative, better electron donor, more positive, better electron acceptor. Then we talk about the order is like this. And I also mentioned about the distance from the beginning to the end. The more distance between the donor and acceptor, the more energy going to generate. And then we talk about the electron transport chain, and we mentioned where is the location. For bacteria, it's in the cell wall system. In eukaryotes, modes, and the fungi, it is at modes and yeast, which means the fungi, it's in the mitochondrial membrane system. And then the electron carrier, we mentioned several of that, which is NAD, NADP, and others. Here we talk about that. Nicotinic adenine dinucleotides, nicotinic adenine dinucleotide, phosphates, fulvine adenine dinucleotides. Now the key things here is for the bacterial cells, the electron carriers, they must be recycled. Otherwise it will not be survived. Then we mentioned about this very important ATP synthesis theory, which is chemosmosis. This is pointed about Peter Mitchell in 1932. So what happened is during the electron transfer, the H, which is proton, accumulated outside of the cell membrane system, generate a potential energy. This potential energy called proton motive force, acronym PMF. Then this one will be activate ATP synthesis, let proton go inside, and then generate ATP. At the same time, there is an activate transport happened and coupled with rotation of the bacterial flagella, which means the bacteria is really going through a movement process. And that is a theory which has got 1959, it's like a Nobel Prize. So it's a breakthrough in the bacterial metabolic area. So here's what we finished from the first class. So today we're going to focus on talk about the left things, and we move on to the lecture 2. So this is the metabolic pathway. We will mention all of these. It could be linear. For example, fermentation, it could be linear. Now fermentation could become complicated, but we usually talk about in this class, it's a linear. It could be branch, which one? Glycolysis, we'll mention real quick. And it could be a circle. It's a cyclic. TCA cycle, citric acid cycle. This you know the metabolic pathway basically have linear branched and cyclic metabolic pathway 3 forms. In order to let the reaction happens quick, we need an enzyme. So here is the thing, what is the enzyme? If we want to say very simple, we had a substrate and will generate is a product. So we want this reaction to happen efficiently and happen quickly, therefore we need an enzyme. Now what is the enzyme? Lots of people say enzyme is a protein. Yes, it is a protein. But enzyme has a different components. Okay, so let's talk about this enzyme. We have apple enzyme. This means it is a protein section. So that's a major section, it's a protein section. Then we have something for help, we call it a cofactor. Now the cofactor depends on how they attach to the protein section. They have a 2 group, which is if it is a firmly attached, we call it a prosthetic group. If it is loosely attached, we call it a cofactor. We call it is a coenzyme. Okay, now, if these add up together, become a hollow enzyme. The most of the important enzyme in a biological system, it is a hollow enzyme. For example, RNA polymerase. Remember what it looks like? Alpha, alpha prime, beta, omega. It's a coenzyme, is that right? This is alpha 2 actually. So we have a 5 units, which is coenzyme. And then we need a factor, sigma factor. It is a coenzyme. So we add up together is the RNA polymerase. Should talk about is a hollow enzyme. But then people just move this out, so become RNA polymerase. So that is an example. So here tells us the enzyme is not only a protein. There is another part, which is a non-protein part. It is also important. Now, because enzyme is major part is a protein, so there is a lot of factors could affect it. Now, we know that pH could be the reason. Temperature could be the reason. Is that right? And also substrate concentration could be the reason, and the product's concentration also could be the factor. That's called feedback infection. So therefore, we talk about the properties of enzyme, composed by protein, high degree of specificity for the substrate, and it's unchanged by the reaction they catalyze, have a high reaction efficiency, require coenzyme for activity, it's heat sensitive. Now, how do you know it is an enzyme? When you see ASE, most likely it is an enzyme. For example, oxidase, catalase, and you see urease. And here, polymerase. When you see ASE, it means an enzyme, okay? This is you already know, so we're gonna talk a little bit quick. What is the major function of the enzyme? You load down the energy required for the reaction. So if you see originally, you need this higher level of the energy. When you put the enzyme, the energy level, free energy level will be decreased. Delta G0, we want them to be less the better. When they less, the reaction will happen more frequently and more spontaneously and more efficiently. So that's the key, is to low decrease required delta G 0 prime. We want them to be close to the negative, or negative as possible. And this is what we already talked about, the structure of the classification of the enzyme. The apple enzyme cofactor and the halo enzyme is the apple enzyme, the cofactors, they combine together. This is something a little bit difficult in your biochemistry area. You need to know the substrate concentration and the velocity, which means the speed, is not linear. It is a curve. And this curve is very similar, kind of like a growth, bacterial growth. Now, why the reason is that? Because at the beginning, when they do this, is using E. coli as a unit. Then something is isolated from the bacteria. So you can see this curve is become flat at the end. Therefore, we come out with a Vvax. What it means? The rate of the product formation when the enzyme is saturated with substrate and operating as fast as possible. Then you will see at the middle there is a Km value. This is always confused by you. That is the substrate concentration required by the enzyme to operate at half its maximum velocity. Why these things are difficult to understanding? Because this curve is a relationship curve, is not a linear curve. Which means you need to have multiple items calculated. Then finally you're going to get X and Y axis the figure. This is different from what we talk about is linear. So that's why these concepts are a little bit to understand, but we're not going to go into detail. We just let you know it is a flat at the end of the day. This is the same thing. Why the reason which is facilitated transport finally will be flat, which means you're not going to be transferred all the time. There is a saturated status will be rich because the enzyme, the substrate concentration, the velocity curve looks like that. Now, there is several models to explain about enzyme activation. This is called the key lock model. It's very simple. You have an enzyme, you have a substrate. They like a key and lock. They put together, they have a reaction happens. Now, what are going to be the conditions affecting enzyme activity? Of course, the temperature. That's the reason why most of the bacteria, the optimal temperature is at 37. Because at 37, same as human being, the bacteria enzyme activity is optimal. This is always also the reason why the pH, usually the neutral pH, is favorable bacterial growth, because that is the enzyme can be a maximized activity. And of course, there is a substrate concentration, products, enzyme concentration, all going to affect it. Then this is we already mentioned. There is a competitive inhibition for that. And we mentioned in our antibiotic section, there is a drug which is called sulfalaminate, which is a sulfur drug. It is a chemical synthesized, but we think it is antibiotics. So originally, you see a PABA. And the PABA, you will see a beta aminobenzoate. This is the substrate bacteria need to synthesize folic acids. Because folic acids, you cannot synthesize by itself. For the human being, we have to take it from the food. For the bacteria, they need a PABA to synthesize. So originally, the substrate and enzyme can be combined together. They are perfectly matched. However, we have a sulfur drug. The sulfur drug is a morphology very similar to the PABA. You can see the top isn't. The bottom is the same, and only the top is different. So, instead of the PABA attached to the enzyme, the sulfur drug could be attached to the enzyme. So the reaction will be stopped. That is one of the ways to stop bacterial growth. But keep in mind that this is the only way to do. However, the function is for static, which means inhibit the bacterial growth. It's not going to killing the bacteria. Because bacteria cannot do PABA, they have lots of the alternative chemicals can be used. Let the reaction happen again. Okay, so this is only for the static, which is inhibit the bacterial growth. Now, lots of the time what happens is that I'm not attacking the target position. I'm going to attack at the back. So once there is an inhibitor attached to the back, they are going to be changed the morphology, and then the reaction cannot happen. This is called allosteric regulation, which is a good example for this reaction. We will mention in this lecture later on. Lacto-opera. This is a typical allosteric regulation. It's very tricky stuff. You maybe heard about that. In other classes, we will talk sometimes next week. We want to make sure you understand about that. And most of the time, the allosteric inhibition is from the end products. So bacteria in their short life cycle, they have to mention it's not overdue. If their end products is too much, please stop. So I'm going to go back to change the enzyme morphology to let them not attach to the substrate to stop the reaction. Okay, so this is a allosteric regulation. Now we end up with a feedback inhibition, which end products could be affect the intermediate substrate, or they could be affect the original substrate. And it could happen in any of the compounds in this chain reaction. Okay, so this is a feedback inhibition. Because I think you already know the enzyme in the other classes, and they're pretty straightforward. So we will be briefly mentioning about here. And then we're going to move on to the lecture. 2 slides. And this will be a key part of the examination 3. It is very important. We want to mention about these one by one. Okay. So first of all, the first slides and the second slide is just a conclusion. Bacteria using energy to survive could be categorized by lots of categories. Based on the carbon sources, they could be categorized autotrophs, heterotrophs. Autotrophs use carbon dioxide as a sole energy resource, carbon resource. Heterotrophs use other organic molecules. So this is based on the carbon source. Based on the energy source, some of the bacteria can use light. We call it a phototrophs. For example, cyanobacteria. Most of the bacteria we talk about in the lab is chemotrophs, use organic, inorganic compounds. The last one is the electron sources. Some of the bacteria use reduced inorganic molecules, we call it lysotrophs, and most of the bacteria in the lab is organic molecules, so we call it organotrophs. However, what is difficult to do? That could be combine everything together. So come up with 5 listed components, which is a combination from carbon source, energy source, and electron source. We don't want to go detail for everything here, but I just want to let you know the bacteria what we did in the lab is the last one, chemo-organoheterotrophs. The carbon source is organic carbon, most likely glucose. Energy source, organic chemicals, same as the carbon source. Electron source, organic donor. And Most of these are non-photosynthetic, which means you using light, using sunlight, they're not going to be survive. That is most of the pathogens, fungi, protists, and archaea. So most of the thing we talk about so far in the lab is the last category. Now then you're going to ask me, how about the first two? The first two basically is the environmental bacteria. They could using light as an energy resource. The most likely is another scientific area of microbiology is water microbiology. We're going to talk about this. How about these two? This is we usually talk about is a sulfur oxidized bacteria. This is the environmental microbiology. When you go Yellowstone, you could see a lot of the environmental microbiology. So in the lab section, we talk about is the last one. Okay, so it used to be this class. We spend in one section talk about photosynthetic bacteria, but we no longer cover it because of time limitation. So we're gonna always in this class we're gonna focus on chemoorganoheterotrophs. This is what we're gonna talk. Okay. Chemoheterotrophs, aerobic respiration, anaerobic respiration, and fermentation. So we want to talk about that. These slides, we could all combine. So what this means? We were using electron transfer, indications of energy transfer. So here is something you need to know. What means fermentation? Fermentation, which means we talk about the electron donor and the electron acceptor. If an electron donor is an organic chemical, and electron acceptor also an organic chemical, this is the best definition called fermentation. Give you a very easy example. Pyruvate goes to lactic acid. Electron donor from organic chemical, electron acceptor is an organic chemical. That's the best definition for fermentation. Now the second one is called respiration. Respiration. What is that? Respiration is a electron donor. I should write E here. Electron donor is an organic chemical. However, the electron acceptor is an inorganic chemical. If this inorganic chemical is oxygen, this is called aerobic respiration. If this inorganic chemical is other, or we say other than oxygen, such as sulfide, nitrite, could be ferric or others. This is called anaerobic respiration. And I want to tell you one thing. Aerobic respiration will generate more energy compared to anaerobic respiration. The reason is the donor and the acceptor, the voltage difference in aerobic respiration is much higher than anaerobic respiration. Okay, so this is the overall picture you need to know. We talk about electron donor and electron acceptor. If electron donor is organic chemical and electron acceptor also organic chemical, we call it fermentation. If the electron donor is organic chemical, let's say glucose. If the electron acceptor is inorganic chemical, let's say oxygen, that is aerobic respiration. If this is inorganic chemical is not oxygen, is others, or we say other than oxygen, this is called anaerobic respiration. We will be focused on, talk about these two, and relate to the energy outcome. Okay, this will be very briefly mentioned, because this one most likely is a little bit related to the environmental bacteria. We don't cover here too much. So let's talk about the detail. This is slides, which is we talk about. You see aerobic respiration, final electron acceptor is oxygen, anaerobic respiration is others. Okay, ATP made by oxidative phosphorylation. This is the same thing. Then we're going to talk about fermentation. So first thing first, the fermentation. First of all, you need to know, there is basically two types of the fermentation. Now, of course, when you talk about the food processing class, we can talk about the whole semester. The fermentation, how we made food products. But basically in this class, we'll talk about two things. What is this guy? Pyruvate. Pyruvate reduced to what is this guy? Lactic acid. Coupled with NADH, oxidize NAD+. This is a key. This is the reason why bacteria can survive in the fermentation. Can you see a whale there? It's a carrier. You can add something. That's why electrocarrier is recycled in the cell membrane system. Pyruvate reduced to, of course, this is called reduced products, okay? Reduced to lactic acid. NADH oxidized, become NAD+. This is we call homo fermentation. This is happens when you have a cheese, when you have yogurts, or any of the dairy products. The major components is lactic acid. That's why you taste acid. No, of course, there's lots of other things. We'll mention real quick. The second, this is a pyruvate. If the pyruvate lose carbon dioxide, will become a very simple chemical. What is this? Acetaldehyde. And then goes here. Also coupled with NADH, reduce, oxidize, become NAD+, then generate what? Where is this oxygen go? Add up here, is that right? How they add up? So become what? CH2OH. What is this? No. Oh, alcohol. Usually we say ethanol. That's 95% ethanol would be used in the lab. What is this called? Heterofermentation. Okay? This is the first thing you need to know, the fermentation. Number 3. In this class, we talk about the energy transfer. How many energy generated? How many ATP generated during the fermentation? Do you learn in your biochemistry class? Very simple. 2 ATPs. Going through what process? Substrate level phosphorylation. Where it is? We'll talk real quick, okay? We'll talk real quick in 5 minutes, 3 minutes maybe. Substrate level phosphorylation. Just 2 ATPs. Here will be the key. Where are the 2 ATP comes? Coupled with glycolysis. Of course, number 5, all very important, you already know. This happens in the absence of oxygen. This is the key. Okay? Now the question here is, how do we know it's coupled with glycolysis? Right here. What happens? Glyceraldehyde 3-phosphates will become 1,3-biphosphoglycerate. This is back NAD plus reduced to NADH2. This is a circle you needed. That's why it coupled right there. Okay? That's why it coupled it right there. So this is some of the things you need to know in this class. Now in my other class, we also talk about this ethanol continue to do fermentation, what's going to happen? Acetic acid, that's what your vinegar comes. You can do a test. You buy a wine, $10 from Kroger. Thanksgiving, you drink a little bit. You left on the bench. Cover it. Well, you need to cover it. You can't expose to the air. Okay, that's a different story because oxygen will come in. You put it there for 3 months. You taste it. It's acid. That's vinegar. But don't use to cook. Okay, that's just a chemical reaction. When really we talk about vinegar, there's a different message to do. That's my another class, for the chemistry class, we talk about how we make some vinegar. But I want to tell you, if you go here, it will become vinegar later on. Okay? So this is something principles regarding the fermentation. So right here you can see it. And we're going to talk about this real quick, okay? This is some fun stuff. Glucose generates pyruvic acids. This is glycolysis. We have a different fermentation products that comes out. Here we just mentioned briefly. If in any chance a bacteria called the probiotic bacteria, what are they going to generate? Do you like Swiss cheese? Taste very acid probiotic acids. And inside you see these holes, what is that? Carbon dioxide made it. Generates Swiss cheese caused by probiotic bacteria. The second one you like it, Aspergillus, Lactobacillus. This streptococcus is not ammonia. That's a specific Streptococcus thermophiles, which means specifically used to the fermentation products will generate lactic acid. Yogurt, soy sauce, cheddar cheese, it's all like that. This is you like heterofermentation, wine and beer, Saccharomyces. Remember the first lab what we did? Candidate albicans, Saccharomyces, cerevisiae. They're called beer yeast. That's heterofermentation, generally carbon dioxide and ethanol. The wine and the beer. Did you go Sephora or go Ultra to buy a nail polish remover? Is that a little bit expensive, $5, $10 for one bottle? It's acetone, is that right? They can remove all those like chemicals. Where they come from? Clostridium. Clostridium is an anaerobic bacteria. It's going through fermentation. It took a little bit of while. So those chemicals, the nail remover not too cheaper. It could be $50 a while. You need maybe a gift card from Thanksgiving from somebody to buy it. That's what it comes from. Acetone generate. It's a key thing to remove the nail. The nail chemicals there, whatever. Last one. Acidobacteria generate acetic acids. This E. coli here is not O157H7. This is E. coli 9037. That's in 9037, people isolated, find it can do during the vinegar manufacturing. That's generated acetic acids, will generate the vinegar. Okay, so this is all is for the aerobic it is for the application of the fermentation. Will generate so many products. We don't want to go into detail here. In my other class, we talk about wine, beer manufacturing, vinegar manufacturing, a little bit about cheese. There are so many cheese talk about that. That could be teaching almost half semester. So this is we just make very briefly here. Now we move on to the big one, aerobic respiration. So we want to talk about some today, some maybe next week, okay? So we're going to remove this part here. This is the fermentation. So we want to talk about aerobic respiration. We need a whole board to explain this to you. You may be learned already. Some of you haven't taken biochemistry class, but more or less you learn something. Okay, when we talk about aerobic respiration, we basically talk about the flow chart. The flow chart you should be very familiar. Energy resource is from an organic chemical, is glucose. Our glucose, we're going through a branch reaction called glycolysis. You can easily pronounce glycolysis if you don't understand lysis glucose. Okay, glycolysis, when you pronounce like this, you can easily say it's a glycolysis. Lysis glucose become what? Pyruvate. This pyruvate could go this way. That is fermentation. Could be either lactic acid bacteria, lactic acid, or ethanol. But major we are talking about here is going through transition step, become acetyl-CoA, and then you have this one is TCA cycle. Now, of course, you know, something go here, pentose phosphate, pentose phosphate pathway. This is an energy resource, a premature material for DNA and RNA. Is that right? Because the 5-carbon sugar comes out, reducing energy is NADPH instead of NADH. That's we don't talk too much here. Okay, I don't talk about too much here. The key line is this. And we will be focused on energy calculation. This will be the key. How many ATPs generate in different scenarios? This will be the key. Okay, so this is the outline. Let's talk about these one by one. First of all, glycolysis. First thing first I want to mention. You learned in your biochemistry class, and we also biology 101, or whatever other classes. We all talk about the glycolysis using E. coli as a unit, as a sample. This is called Epstein-Meyerhof pathway. It's a standard pathway. But I want to tell you one thing, in the bacterial area, this is not the only pathway. If the soil, environmental, water microbiology, it's called Epstein-Dolohov pathway. It's another one. Basically, this beginning is similar, the branch is different. Okay, so just want to be aware, this is, when we talk about a bacteria, this is the standards, but not say every bacteria is like that. Otherwise, it's so easy to control. Epstein-Meyerhof pathway is a standard we use for teaching. This is what it looks like. So let's talk about these. When you talk about learn the glycolysis, the first time you learn, or you see these things, glucose, glucose 6-phosphate, fructose, blah, 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 okay, seems like too much. I would say when you learn, you step by step. The first thing you learn, you need to know this is a glucose. Going through a branch, generator end product is pyruvate. This is the first step. And how many ATP generated? Now, when we talk about here is an individual cycle. In the absence of oxygen, only 2 ATPs generated through substrate level phosphorylation. This is exactly what we talk about fermentation, where that comes from. And all of the chemical textbook, it's a little bit confusing. They always say the glycolysis is only 2 ATPs comes out because this is in the absence of oxygen. And I think it's an individual cycle. And you also need to know, this single one here is an investment. Is an investment stage. 6-carbon stage we also have. This is like you buy a house, buy a stock. Or maybe by Powerball or something. You need to pay some money. It's an investment. This is the one you will get something. Is that right? Which means you gaining ATPs. And this is 3-carbon phase. This is the first thing. Before you go, you need to know. If you don't know about this big picture, then you go all the others, you just remember. So the more you remember, the more you reading, the more you're confusing. So that's the big picture you need to know. So then let's go with these one by one. Okay. First of all, no matter what happened, I believe in the university, you should know what is glucose structure. Okay? This is glucose structure. This is D-glucose. Now, here, the carbon could be up and down. That's L-glucose. That's a standard glucose. And you should also know, this is a Fischer configuration. Remember, you learned about 3 different configurations, all right? One is an open ring, is a Fischer, another one is chair. Chair is more confusing you, like a couple of match briquettes, like it's more confusing. So we were using a Fischer configuration right here. So this is the first one, glucose. Okay, the next one, where are we gonna go? Glucose 6-phosphates. Okay, I all use P representing phosphates. Okay, I'm not gonna go much, write all the phosphates, just use a P, okay? Now, what is glucose 6-phosphates? Then you need to know the position, is that right? Carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. So what is glucose 6-phosphates? Others are all exactly the same. Exactly the same. You can just draw it. This is what I teach you in the exam, okay? You get a glucose, you get 1 point, you get a second point, which is glucose 6-phosphates. Where is the position? Right here. That's glucose 6-phosphates. Okay, now the question. Where is this phosphates comes from? Where does phosphates come from? ATP hydrolyzed become ADP and release our phosphates. Okay? What is the enzyme for this one? Hexo-nice, is that right? What do you call it in the biochemistry? Hexokinase. Okay, here we want to mention. What you learned in your biochemistry class, hexokinase, is for eukaryotes, yeast, and the modes in this class. How they generate in the bacteria? Do you remember group translocation? In the bacteria, this will be the major function. Bacteria doesn't go this too complicated. When you create the hexokinase is all eukaryotes. Bacteria going through group translocation directly become glucose 6-phosphates because speed up the ways it could happen more quickly. Okay, now next one. Just like that, okay? When you see this type of the structure, you're not going to use too much. This is very stable from the chemical standpoint. So they have to become glucose 6-phosphates. What it looks like. This is a very magic stuff, if you really understand. I'm telling you. Okay, this is OH. Okay. This is H here. CH2O. When you change to this